So, howdy, and welcome to lab report number 3, on which was the staining lab, on which we did simple staining, gram staining, acid fast staining, capsule staining, and endospore staining. This was performed by Group 4, which is made by Vianka Sanchez and Lucas Perez. That's me. And this was done for the BTPV 4 or 5 class or microbiology class for the fall 2024 semester. So, for the introduction, we would be going over what we just did in the lab for these past weeks, which were the lab stainings. And the lab stainings are important because it helps us to classify the microorganisms based on their cellular characteristics. And for this past lab, we did 5 different stainings, which was the simple staining, which help us to see the size and the arrangement of the bacteria. The gram staining, which help us to see if they're gram positive or gram negative. The acid fasting staining, which help us to identify bacteria with waxy cell walls. The capsule staining, which help us highlight protective capsules around the bacteria. And the endospore staining, which detects dormant endospores in bacteria. And for all of these slides, for all the pictures, we did smearing techniques, which we're gonna talk more on the next slide for the methods. And that smearing technique is the almost exact same thing as the exact same technique for all of these procedures.
The materials for this lab included these following cultures: Staphylococcus aureus, Bacillus cereus, Salmonella typhimurium, Klebsiella pneumoniae, which was used for the capsule stain on blood agar plates, and then our mixed culture, which was Staphylococcus aureus and E. coli, and that was in a tryptic soy broth. We also used a wax pencil, glass slides, a microscope, inoculating loops, a Bunsen burner, bibulous paper, and our stains, which were crystal violet, simple stain, and gram stain. Our gram iodine mordant, which was used for our gram stain, our decolorizer, and our acid alcohol decolorizer. These were used for a gram stain, and then saffron, which was used for our gram stain, which was the gram stain counter stain. Quinone carbofusion stain, which was used for the acid fast dye. Methylene blue or brilliant green, 20% copper sulfate, and malachite green.
So for our methods, beforehand, before doing any of the staining, everything, we had to prepare a slide. So for that, we had to clean a slide. Once we have the slide clean with the wipes and the chemical that we use to clean it, we do a little circle with a wax pencil. With that wax pencil, we put a circle, and on that circle, we put a drop on it. On the other side, we don't want the bacteria to be on the same side of the circle. We're gonna put a bacteria on the other side, and we're gonna dilute it with a little bit of water so it mixes. We let it dry, we heat fix it, and we have the slide ready to be stained. Once we have all that done, we move to simple staining, on which we use a dye. In this case, we use crystal violet to give a little bit of color so we can see the shape and arrangement of the bacteria. Next, we have the gram staining, on which we use crystal violet as our main stain, followed by iodine, then followed by a decolorizer, and finally we use saffron as a counter stain. On this process, the crystal violet would stain to the gram positive and the saffron would stain to the gram negative. So giving a gram positive a purple color and a gram negative a pink color. For the third step, we have the acid fasting stain, on which one we use the carbon fusion and heat. Uses a acid alcohol to decolorize, and an acid fasting bacteria will stay red while the other one will turn blue with a counter stain. Also, we have the capsule staining, which uses a negative stain like Indian ink or any other type of stainings to outline the bacteria, showing the clear capsule around them. Lastly, we have the endospore staining, with magellane green as their main stain, using heat to help it penetrate the endospores. Rinse, then counterstain with saffron. Give So for the first of results, we have the simple staining, on which we had four bacteria. The four bacteria were B series, S Aeres, unknown 104, and unknown 204. All the bacteria are gonna look the same color as the main dye that we use. In this case was crystal violet. So it doesn't matter if they're gram negative or gram positive, we're still gonna have purple in them because of the crystal violet that we use. This stain is not to see the type of bacteria there are, it's just to see shape, size, and arrangement. We can see that our B series has some rod shape that appears single cells or in short change. If you zoom in, you can see more a little bit of what I'm talking about. Also, the S areas, you can see that they're round cocci that form clusters resembling some type of like grape structure. And you can also see that our unknown 104, it also has rod shaped uh, seven areas that it might look like single cells, but you can see some of them make change changes, and some other look like independent cells. Also, we can see that our unknown 204 appear very similar to S areas, and they're both round clusters that combine multiple like colonies into like a big change slash cluster. So for the second set of results, we have gram staining. So for the gram staining, on these pictures we have unknown 204 and unknown 104. You can see how our unknown 104 appears purple rods, and the purple color on gram staining shows us that the bacteria is a gram positive bacteria. But on the other hand, we can see that our unknown 204 appears as pink cocci, indicating that it's gram negative bacteria. Something that I would like to point out about this slide is that during the process we had some complications on the staining. This is actually the second try that we had for unknowns, but we were supposed to have K summonite and a mixed culture of S. aureus and E. coli, but unfortunately due to poor technique and shortage of time, we couldn't get those, and we had to mainly focus on our unknowns. And that's the main reason why we don't have any of those, because of poor technique and resulting in unacceptable results. For the second set of for third set of results, we have the ass fasting staining, and the ass fasting staining mainly divides into two, negative and positive, which mean that if it presents the waxy cell wall or if it doesn't present the waxy cell wall. And for this test, we had an already prepared slide for us, so it means that we didn't do the heat fixing, we didn't do the smearing, we didn't do anything. The slide was already prepared for us, and it was divided into two sides. One of the sides was with a positive bacteria, which was the mycobacterium, and the other side was with a negative, which was the an unknown bacteria that we didn't know about. And you can see how on the positive, it gives us like that, like kind of like pink reddish color, because of the retention of the carbon fusion. You can see how we can see like those type of like rods as like strings if you zoomed in a little. And also on the other side, we have the non-fasting acid fasting bacteria, which is the unknown one. You can see how most of the color like didn't stain to it. It's kind of like fainted, but that shows us that that bacteria didn't have this the waxy cell wall, and that's why it looks like that, more fainted. But if you zoom in, you're able to actually see the shape of the bacteria. For third, so for our fourth set of results, we had the capsule staining. And the capsule staining is mainly done to highlight the protective capsule around the bacteria. And in this case, the bacteria that we were using was K. pseumonite. The K. pseumonite shows a clear halo surrounding the bacteria cell. The cell is stained as a purple bluish color, and the capsule remains unstained. You can see how the shape of the K. pseumonite It looks like a little like rods, and we didn't stop just with the regular process. We went a little bit over it, and on the same slide, we applied 2% copper sulfate, which works as a decolorizing agent for the capsule. This and the cells will be stained purple blue, and the capsule still remain a lighter color. That's why on the left side you cannot see the capsule shape that clear, but on the right side you could see more of the shape of the cell, the bacterial cell capsule. So for our last set of results, we had the endospore staining, which is mainly done to detect the dormant endospores in bacteria. In an endospore staining, the endospore will stay in green color while the vegetative cell will stain in pink/red color. This is because of a stain that we use that is called methylene green, which is the primary stain, and then followed by heated penetration, and all the process of the counter stain, the saffron, um, and that gives the stain its color. But unfortunately, for this slide, we couldn't get the results that we wanted. Mainly, our main hypothesis of it is because of our dyeing technique. We feel like it's not appropriate. It could also be the heat fixing, but we haven't done any more testings on it. But those are our two main hypotheses: the dyeing and the heat fixing technique. But
Simple staining uses a basic dye to enhance the visibility of the bacterial cells under a microscope, and the stain depends on the dye that's used. So, since we used crystal violet, the cells were stained purple. The Bacillus series appeared as rod shapes, both single or in short chains, and the Staphylococcus aureus were round cocci, which form like grape-like clusters. Our unknown 104 also displayed a rod shape but only as single cells, and then our unknown 204 resembled S. aureus, which appeared as the cocci, the round cocci forming grape-like clusters. The Gram stain classifies bacteria into two groups, which is gram-positive and gram-negative. Gram-positive retain the crystal violet due to their thick peptoglycan layer, while the gram-negative bacteria do not. Instead, they take up the counterstained saffron and they appear pink. So our unknown 104 appeared as purple rods, which indicates that it's gram positive, and our unknown 204 showed pink cocci, which means it's gram negative. During the staining process, we also attempted to stain Klebsiella pneumoniae and a mixed culture of Staphylococcus aureus and E. coli, but due to our poor technique, the results were unsatisfactory, and we believe our main issue was the dyes. We found that the problem was the amount of bacteria we were smearing in the slide. We were putting too much alongside bad heat fixing. Simple. The acid-fast stain identifies bacteria with the waxy cell wall. Acid-fast bacteria maintain the major stain, which is carbofusation, and it gives them a bright red pinkish color, while non-acid-fast bacteria absorb the methylene blue counterstain, so they stain blue. We used a microbacterium slide that was already prepared, and one side of the slide had the acid fast positive microbacterium, and the other side contained the unidentified non acid fast organism. The capsule stain stains the protective gelatinous layer or the capsule that surrounds the bacteria. In this stain, the bacterial cells appear a dark purple while the capsule remains clear and colorless. For the Klebsiella pneumoniae, the capsule was visible as a clear halo around the purple bluish stained cells, and the 2% copper sulfate serves as the decolorizing agent for the capsule, so it leaves the cells stained purple or blue while the capsule remains unstained.